Hello, my name is Kelly, and today I will be presenting the differential expression of appetite regulating genes in avian models of anorexia and obesity. The background information, chicks that were selected for low or high juvenile body weights for more than 57 generations to gain insight on appetite regulation were used. These chicks were 5 day old and they were either fasted or they were going to be provided with food. Then the mRNA abundance of various factors and appetite associated nuclei in the hypothalamus such as the lateral hypothalamus, paraventricular nucleus, ventromedial hypothalamus, dorsomedial nucleus, and arcuate nucleus were measured. And there's two pathways for hunger versus satiety. There's the orexigenic pathway, which deals with neuropeptide Y and AGRP. And then there's an orexigenic pathway, which deals with POMC and CART. So let's look at the lateral hypothalamus, which basically is the part of the brain that is responsible for hunger, versus the ventromedial hypothalamus is the part of the brain that's going to be responsible for stopping hunger. And these are different figures that explain the concept. There's the VMH again, and here's another figure that talks about the pathway that we will look into more in details. And there's the DMN. Here's another figure that talks about its pathway. The arcuate nucleus basically has neurons that sense the feed or fasted state, and they regulate hunger. So the agouti related protein neurons in the arcuate nucleus are gonna be stimulated by fasting, and once activated, they're going to rapidly drive hunger. And here's the pathway depicted in some more background information on how leptin is going to inhibit neuropeptide Y and AGRP, and then leptin is going to stimulate POMC and CART. And here's alpha melanocyte stimulating hormone. Basically, it's going to be made by POMC neurons in the brain, where it's going to act to suppress appetite. And so in some cases, extreme obesity has been traced to mutations in the brain receptor of alpha MSH. And then we have here POMC, which can be cleaved to make MSH, corticotropin, and beta endorphins. And we have neuropeptide Y, which is a 36 amino acid neuropeptide that's going to act as a neurotransmitter in the brain and in the autonomic nervous system of humans. And it's going to be an orexigenic agent that is presumed to play a leading role in regulating eating behavior, as I explained earlier. So the materials and methods, the 5-day-old chicks were used, and they were randomly divided into 4 groups: LWS fed, LWS fasted, HWS fed, and HWS fasted. The duration of the fast was 180 minutes, and the samples of individual nucleus were collected using the sterile disposable biopsy instruments. The punches were immediately submerged in RNA lysis buffer. So punches were vortexed and incubated, and the RNA was assessed by spectrophotometry. cDNA was made, and then the primers were used for real-time PCR, which was performed under the conditions of 95 degrees, 90 degrees, 60 degrees, and then 90 degrees with varying seconds and different cycles. What is PCR? PCR is a technique used in the lab to make millions of copies of a particular section of DNA, and it was first developed in 1980s. What is cDNA? cDNA is basically when we're going from RNA to DNA via reverse transcription, and it enables researchers to study RNA with the same molecular approaches used for DNA investigation. So the cDNA generated by reverse transcription is going to be amplified with PCR like we saw before, and these are the PCR primers that were used. And now let's look at the statistical analysis that basically included the main effect of genetic line, feeding state, and their interaction. and that's how we analyzed it as statistics. So let's look at the mRNA abundance in DMN. So the mRNA abundance of neuropeptide Y and MC3R was greater in LWS than in HWS in the DMN. And the reason for that is that there was an interaction of genetic line and fasting treatment on the expression of MC3R in the DMN, where mRNA abundance was increased by fasting in LWH but not in HWS. Um, for the mRNA abundance in LHA, and the LHA expression of neuropeptide Y receptor subtype 1 was greater in the fasted versus fed state, whereas CFOS was greater in fed than fasted chicks. So there's a two-way interaction on expression of mesotocin receptor, where expression was greater in HWS than LWS in the fed state, but similar between the lines in the fasted state. Expression of neuropeptide Y receptor subtype 5 and orexin was greater in LWS than HWS in LHA. Results for mRNA abundance in the VMH. C-false mRNA was greater in fasted than fed chicks, and expression of orexin receptor 2 and LEPR was greater in LWS than HWS chicks, as seen in Table 5. For mRNA abundance in PVN, the mRNA abundance of NPYR5 and ghrelin was greater in fed than fasted chicks, as seen in Table 4. So expression of NPYR1, MC4R, and CART was greater in HWS than LWS, whereas NPYR5 and MT was greater in LWS than HWS in the PVN. As seen at Table 4. For the arcuate nucleus in the neuropeptide Y receptor subtype 2, mRNA was greater in fed than fasted chicks. So there was an interaction of line and treatment in neuropeptide Y and CRF receptor subtype 2. For the discussion, interaction of genetic line and fasting on mRNA abundance is shown in Figure A at the top left. MC3R, which is depicted in the dorsomedial nucleus. For Figure B, there's mesotocin receptor and the LMH. For Figure C, there's neuropeptide Y in the arcuate nucleus. Figure D, there's corticotrophin releasing factor receptor in the arcuate nucleus of the hypothalamus. And so we can see that basically at 5 days post-hatching, the chicks from lines selected for low or high body weight were provided continuous access to food or were either fasted for 180 days, and hypothalamic nuclei were isolated for measuring mRNA abundance by real-time PCR, as these figures show basically what we explain in. So the LWS became anorexic and the HWS became obese. Fasting increased melanocortin receptor 3 expression in DMN and neuropeptide Y in the ARC of the LWS, but not HWS chicks. So expression of neuropeptide Y was greater in LWS than the HWS, and the DMN neuropeptide Y receptor subtype 5 mRNA was greater in LWS than HWS, and the LHA, PVN, and arcuate nucleus. So expression of orexin was greater in LWS than HWS and the LHA. So the results show that the orexigenic tone in LWS overrides orexigenic factors such as neuropeptide Y and AGRP that were more highly expressed in LWS than HWS in several nuclei. For critique of the study, in the future we can compare chickens with other vertebrates, as the study had an interspecies comparison between fed and fasted chicks. We can also do that with other species. Also, we can explore whether or not different sexes of chickens could have a difference in the study. Overall, this paper was well organized. The concepts are made clear to the reader, and there's enough information for another researcher to replicate the study. These are my references, and thank you very much.